So, welcome to the second part for today. This will be about image analysis now. So, this morning you had a lecture about how to acquire the images, all the techniques that are related to it, and the things that you need to know and take care of about getting actually the image properly done. What systems are available to do this. But now we assume this is done. You have the image from the microscope, and now you want to analyze it, but with focus on JIPipe more. So, which will JIPipe is a more automated analysis platform. So you can of course open any image that you acquired from your microscope in an image editor, an image tool, and then look at it, and then of course do the analysis manually. I did this enough with myself when I had some practical course at the lab counting things manually. And here I have a small example that what, for example, could be produced by a confocal microscope. You have image here, a confrontation assay, in our case two-dimensional only, where you have 4 channels. The red one contains macrophages, green contains fungal spores, blue fungal spores that are outside with a different staining, and then transmitted light. And then the task there from this study was to count which spores are inside the macrophages and keep track of their color, because then we can know which ones are really inside and which ones are outside. So you have to do some overlapping and then see if it's really in a cell or not. And then make use, for example, of this red channel. And here's an example of such a segmentation. This was done automatically, but of course you can imagine you can do this in something that can annotate your data, and then you do it manually that way. And as a side note, this was actually done, a lot of it manually by the person who did this, so counting their cells. And of course, you can imagine that mostly you might think it is straightforward to do a manual analysis, but it's actually not that straightforward often, because you have all these weird issues going on a bit with these images, where it's a bit not entirely, you're not entirely sure what actually is happening there, and if it's that or this. Example, I picked out some here. So how many spores are those? This is unfortunately directly at the tiling border. So which one are spores, which ones are some debris that has been left over? What is with this weird tiling border where the stitching did not was not correctly applied? How many spores are there? And then sometimes you have some weird decisions to make. Okay, is it inside or outside? It's hard to see on the screen here, but there's like this red outline surrounding it. Where do you put the cutoff? When is it inside? When is it outside? Is this here a spore or not? I don't know. I'm not the expert, but the expert also has to decide it. And here again, we have a problem where it's not so easy to determine if it's inside or outside, because here this whole part is basically red-ish. Do you put the cutoff? And so now you can do this and analyze it, but you let someone else do it, and then you will get a completely different result out of that. And the worst thing is, it makes a difference if you do it in the morning when you are fresh and you got up and you had good sleep, versus you are at the evening at 10 p.m. and then you just wanted to get it done. You will get different results. So this is not the ideal thing, that we have these researcher biases going on. And then also, this is of course not the image, this is just a cutoff. The whole image looks like that. And then even you have like a whole directory of those. So have fun to count this manually. I let this done by you. I will not do this. I think Mohammed, who did this originally, he took like 8 months or so, counting it manually.
He spent most of it mastered.
So if you want to do this, then you can do this, but not me. And I hope you also don't. That's why you're here. So you're learning image analysis, and the computer do it for us. So to summarize, we have here the manual analysis. It starts with researcher bias. You have variability where it's hard to make decisions, and you have hidden factors on the decisions. When is it inside? When is it outside? Where is it written down? Who decides it? It's somewhere here in your brain, and it's not written out in a formula or something or in code. But then you have, as alternative, automated analysis methods, or at least semi-automated analysis methods that take some input from you. The computer will do it for you. And they mostly have no variability. There are some algorithms that have some random initialization, but let's ignore this. The computer will not be always correct with what it finds, depending on how you set up the algorithm. Sometimes you need some testing out different parameters, trying different approaches. But the thing is with the computer is, you can identify where it's wrong. You can go through the pipeline and then see, okay, there it fails. Then you can do the corrections there. And then hopefully it works afterwards. And you have, of course, there are then no hidden factors, or it's less hidden factors. What is going on, you can look up, you can go into the code, at least for open source software, how does the algorithm work, what step is applied, at which time and which condition. You can usually explain all the criteria, except AI things. AI is a bit, this is an active field of research, how AI actually works and what does it see, but it still works very nicely, so everyone uses it. And also another benefit, of course, the computer is fast, and you can click some start button, let it run overnight, you can sleep and it does whatever. And then later, next morning, you come back and then it's done. Hopefully everything is correct. There's of course many tools that you can use, and in previous years, or still there are some that are very popular. There are many commercial tools that you can use. I've listed here some. This is actually from the older version of this lecture that I carried over from Sultan. So we have Imaris, which we still have here. It's still one of the best 3D visualization tools. Velocity, MATLAB is also often used. There are some recent conference who use MATLAB. Huygens, which does this deconvolution step. And we still also have it here because it's basically the gold standard of deconvolution. And you see here the nice price tags that are attached to these tools that are highly variable, and you have to quote the companies what you actually have to pay them. So these are approximations. For example, Huygens likes to charge based on what hardware you have. So if you have fancier hardware, they want to have more money for that. But you get a nice ceramic house every year if you are a customer. So this hopefully helps out with offsetting the cost. So what you end up using if you not want to get poor is using open source software that is getting more and more traction, and it's getting better over time even. There's common tools like Cell Profiler, which is very popular for analyzing cells, and it has many features. IC and NIME, okay, NIME less so, but IC has focused itself very much on 3D data. And then the topic of our course here is ImageJ or FIGGE, and then JIPipe, which is related to that and based on ImageJ and FIGGE. But before we go into software and do something in JIPipe, we have to establish some theoretical basis. For example, what this even is image. How is it represented in computers? important concepts like the dynamic range, the bit depth, which file formats you please choose to save it out, otherwise you will have problems in the end. And then the steps how to do actually the image analysis, because these things are all very universal and you can apply them to many different tools. If you, for example, jump later onto Python or whatever new tool is around, these are all based on these things, and you can apply these steps that you have learned here. So first, what is an image even? And most basic thing and most commonly used one, for us at least, is an image is basically a grid of pixels. And each pixel, I zoomed here in, it has a value that you see here is some grayscale value, but the computer has finite memory. It is not like universe infinite, so it has some kind of limit what values can be represented in each pixel. So for example, if here, an example of an image where it's basically its limits of its values and what is possible to be displayed in there is determined by its so-called data type. And that's a very important concept, this data type, to understand. So basically each pixel stores a specific fixed data type inside that in our case can be integers, can be real numbers, so something with a dot, or it can be colors. And then in my example, I chose an image that stores an integer, an integral number, so one that has no comma or something. And then if I pick out a value here, then this one is value 10. This one here is 201. You can do this, for example, in ImageJ. You can go there with the mouse, and then it says, okay, this is the value. And then there's its related concept. It's the so-called bit depth. So, as mentioned, nothing is here infinite. Each pixel has only that much storage. And so it needs some space to store the information, and this can be only finite. And this is basically, the bit depth is basically related to what space you have available in each single pixel to store the values. And computers like that as well, so they can handle that fast. And it's called bit depth because computers work in binary. The smallest unit it works on is a bit, so 0 or 1. And you will see soon examples of what I mean with that. So let's look at some of these bit depths that are available. Imagine if I picked out one of these pixels and then I showed basically what's inside there in binary code. So if we have a 1-bit image, the pixel, if I print out basically the individual bits or memory that is in there, it would look like that here. 1-bit image, there's only one space with 1 bit. This can be only 0 or 1, and then nothing else, because there is literally no space afterwards. I can tell the computer to make me a 2-bit image. There I get 2 spaces, and this can encode integer numbers up from 0 to including 3. So 0 is everything 0, and then this one, and then 1 would be 0, 1, depending on how you read it, which way around. And then it continues to go on to 2 and then 3. But then we are run out of space. And then we can continue with that. We can have a 4-bit image where we can have more values that can be represented by a single bit inside. And then 8-bit images that have 8 bits, and then we can go up to 255. But that doesn't mean that we have this range. We can also decide to tell the computer, okay, please take me from these whole 8 bits, half of them, and always say, okay, I say the first 4 bits, they encode something different than the other half of 4 bits. bits. So it's basically now a two-channel image where we have two from 0 to 15 numbers that can be represented in one pixel. And this will be a bit important later on. This is just an example. No one does it like that with 8 bits. Or we can say we have a so-called 32-bit image, a floating point. And there you see something interesting. I said there's no infinite, but actually it's possible with some tricks to represent something that's infinite if you are getting, if you allow that the precision goes down the further you are away from 0. But this will be explained in another slide a bit better. So this is a 32-bit image where these individual bits are split up in a very specific way, where the first one is called a sign, and then you have an exponent and then a fraction. This is basically this IEEE standard of representing floating point numbers. You can look it up on Wikipedia, how it works and why it exactly is like that. But for this one, it's just important that we can do that. We can split up these 32 bits that we have and then assign them a new meaning instead of just saying, okay, this is a 0 to 255 value. For 32-bit, it would be like 0 to 4 million something. So, okay, summarize it a bit. Someone's stuff dropped, very good. Bit depth is basically how much memory each pixel is allowed to have. And then this whole splitting it apart, this is basically part of its data type. So how to interpret the bits, should we take them as is and then say, okay, it's from 0 to 255, or is it 2 times 4 bits and then we have 2 channels? And these are very closely related to each other. And you will encounter mostly those here. These are the ones that ImageJ supports, where you have the 8-bit image. They just call it 8-bit image, but it actually is an 8-bit image with integer data type, and it has a value range from 0 to 255. Then are also 16-bit images that just say, okay, all the values are already interpreted as integers. And these are very common in microscopy. So many microscopes produce such 16-bit images because you have a higher dynamic range than those. And then also ones that you are very common with all the time. You're seeing it in front of you. The 24-bit image. That's basically 3 times 8-bit integers. So we take the 24 bits, split them up into 8 bits. And this is how RGB values are encoded. This is how color is represented in a pixel. And then the floating point values that are theoretically from negative infinity to positive infinity, but the further away you get from the 0, the less precise they get. With funny effects, so you can read up on that. So these are the ones that ImageJ has, and these are the most common ones. There are also some special intermediate ones, like in between 8 and 16, there you will find 8-bit integer or 12-bit integer, but ImageJ doesn't really handle them and auto-converts them to 16-bit. Although you see them in their calibration tool as like a setting. And as I talked about colors, there's also some additional concept that is attached to it, color spaces. So the most common one you see currently even is RGB, because if you zoom into your screen that you're sitting at, then it looks like that, or some variation of this pattern. Red, of course, this one is not RGB, it's red. It is red, green, blue, and this is basically how most of our screens work, 100% colors. But this doesn't mean this is the only way. There are other color spaces that you can use. So if you look at this in the bit level, again, just to illustrate it, 24-bit image, and then instead of saying that all these 24-bits are used to represent one number, we split them up into 3 numbers, each representing one of these channels. So there are others that other ways how you can interpret this, and these I show you later at some point, because I will first go to false colors, which are related to color spaces. So you will encounter them usually in ImageJ and in any image tool, and it's important to know the difference between the real colors or the real values versus the false colors. So you can go into any image tool and say, okay, this is here my image, it looks a bit boring. I can tell the tool to make it pretty colorful. This is usually very helpful if you want to present something to media. They love it when it's colorful. So, but this is just something the computer or the image viewer will do for you when you're looking at it. Actually, the real image looks like that in the background. It's still grayscale, but when it's rendering it out onto your screen, it applies a coloring map that is maps each intensity to a different color. I have a few listed here. This basically assigns an RGB color to a grayscale value without changing the image, just when you're displaying it. And here are some nice colorful ones that might or might not be helpful depending on what kind of data you're looking at. For example, JIPipe likes to use this viridis pattern as default in Python also. And you can, an ImageJ comes with loads of them. They're in a menu, LUT, and JIPipe also has them. So you can play around, make some visualizations, which is also part of the of some exercises to make such visualizations with nice colors. So, as we talked about a bit about these colors, why should I care about this abstract image and data type and everything, and splitting the bits? You see them in ImageJ and then they do something and they work somewhat, unless they don't. So why should you know about this? Because when you output things from your microscope and you go to the settings there, you can absolutely mess this up and then make the analysis completely unusable later on, or make it very, very hard. I had actually a recent case where, no, this was a different one. But there were cases where people saved it out in the wrong format, for example. So there are some microscope software, I think, where you can tell the bit depth that it saves out. And some people might think, oh, the 8-bit, it makes a smaller file, then it doesn't use so much on the hard drive. You will lose information that is then gone, and it will never be able to return. It's like in a black hole, it's gone forever. And you don't want this, because this can mess with your analysis later on when you need this information to, for example, detect the structures as you want. And this is something that, because bit depth costs memory, then we have experienced this. People choose the wrong file formats to save a few megabytes on the hard drive. And that's why, and this makes it very complicated, because there are many file formats that are around that you can choose later on. So you find everywhere like TIFF and OE-TIFF and PNG and JPEGs, and people use GIF files and then bitmaps and then Photoshop documents and so on. Again, to emphasize, you choose the wrong one to convert it, and then because it looks nice or it's nice and small, you will lose information that is then gone, especially if you did the original file, and then in the image analysis you can sometimes do something, but it's not hopeful. That's why I made this chart. It's useful for you maybe, with these image file formats and where you should use them. So we have here GIF and JPEG. These are very compressed formats that are suitable for putting them on websites or something. So they are very, very small. They make weird artifacts. GIF for grayscale is fine, but colored GIFs, they cannot represent the full color range and then make weird patterns. JPEG makes these famous blocky artifacts. This is fine for a website. For the analysis, you will see these blocks and then they do weird things with your statistics. For publications, bitmap is maybe possible, although they like now for PNGs or PDFs or something. So PNGs are mostly that. So they are similar to JPEGs, but not so small, but they have a different way of compressing the information, so they don't make these weird blocks. When you're making contents and figures for later to be published, use whatever from these native formats that are available from these tools that you use. Photoshop's PSD format. The GIMP image editor has XCF. Inkscape has SVG. You keep them around because then you can edit all your things again if you need to change the figure. And then now the scientific category. And these are important that you keep them around. The top one is the native format that your microscope will produce. So, for example, in size microscopes, they will produce CZI formats. These contain the most information of all because they're literally from the microscope manufacturer and they store all kinds of metadata inside, including what lasers you have used and the colors and so on. I've received data now for your exercise you will be hopefully looking forward to, where I did not know the channels, but I could look this information up because it's stored in a native file format. And it says, okay, channel this is this dye. And then other things, the physical sizes of each pixel, so how many microns a pixel is actually in real world coordinates. Then you have it going a step down, but not too much. These international standardized formats, OM-ETIFF and OM-ISAR, although OM-ISAR is quite new. These are basically these people who make these formats, they work directly together with these microscope manufacturers that all their metadata is carried over into this format. So it's also a valid way. These native formats might contain a bit more information, but these are also usually fine, this OM-ETIFF or ISAR, to save your data. And these are also the formats that are related to this image database, Omero, where you can upload images on the cloud, which is made for scientific purposes. And then a step down from that is this normative format, which doesn't have the strong metadata information as OM-ETIFF. ImageJ has, for example, its own variant where it stores some information, but it's all over the place and useful for maybe for intermediate results or for test data. Ideally, stay here with these two. So you can read these in and then maybe save those out, because saving these native formats out, none of these tools can usually do. So, and then something else that is important is these can all handle 3D data or higher dimensional data. Especially TAR, it can handle up to infinite dimensions, and TIFF and the native formats. The native formats I don't know, but TIFF definitely can handle 5-dimensional data. And that's why we look at it now, because this, you can create 3-dimensional data, but it's good to know how the computer actually handles those and how they are saved. So you get sometimes 4-dimensional, 5-dimensional, whatever dimensional images, and there are different ways how you can store them. And you can imagine, okay, can I store them as big, large chunk, like a cube or hypercube? You can all index this. or do it simply by having multiple 2D images and then they just stack them on top for a 3D image. Or you store them as multiple files in a folder or one file, you can choose that. And this is actually here, these multiple 2D images, this is the way how TIFF handles the storage of 3D or higher dimensional data. So these are called planes in TIFF. And in normal TIFF files, not the OME ones, they are a bit more flexible. They are stored in one single file. And it's just ensured that these planes all have the same data type and the bit depth, and the same bit depth. Otherwise, there would be some weird things going on and it's not optimal. And then each plane basically has a little attached metadata about its location in the so-called hyperstack. So, and this metadata specifically is a channel, depth, and frame. And this channel thing is funny, I will mention it later on. So for example, I have here one that I showed it always on the left side. This is a set stack. So we are going only over the 3D axis. And then you see here that the C changes, but the channel and the time axis does not change over these slices. And of course, there can't be duplicates, then it would be not known which one you want to choose, and of course no gaps between those, so you cannot just default C equals 4. You need to have something in there. Otherwise the format doesn't work and then it's not optimal how it saves it.
Sorry?
Yes.
One question for the slide before. Does that stand for the Z?
Yes, this
Does
is
it stand
usually...
for the location? Does it stand for the location of the image?
So this is... the naming of these is a bit, you have to get used to how these TIFF things load. This is basically, you can take it out. This is like an image, like a PNG that sits somewhere. So if you take one of these slides out, and then basically how a TIFF works is it contains many of those and then it assigns, it's like a metadata. Okay, this is now at C1, Z1 and T1, and then internally it handles it somehow. So it's like a file system in a way, basically. So these names can be a bit difficult or a bit
confusing.
Yeah, it can be confusing, especially because depending on the library. So, for example, you have Python's loading library. They will take this whole C stack and then just make a 3D cube out of that because it knows how to handle those. Or if you have like more dimensions, Python will handle it. But ImageJ can handle only theoretically 2D images, so it organizes them and you can go plane by plane, like when you go through file by file. So this is something to get used to in this tip. And this is not even the maximum level of confusion. So because there's this nice confusion that they established from TIFF side, channel versus channel confusion. So I mentioned by explaining RGB colors, we have here these red, green, and blue channels. These are channels. These are also channels. And they're both called channel, but they are different concepts. The funny thing is that you can have a TIFF file that has multiple TIFF channels that themselves store RGB color images that then have channels. Each one has 3. Although not all tools can handle this. Python especially does. It can handle either. So if you give it the RGB, it will convert it basically into this internally in some way. And if it gets both, then it will do something. So you get these weird things. ImageJ, fortunately, can handle this. So ImageJ can handle a TIFF that is RGB colored and has multiple channels and Z axis and time axis, absolutely fine. When you're working with Python, be careful with those. Very careful. OME TIFF is also a thing. It's a bit weird how they handle these things. It comes from how they implemented it. And then to add even more complexity with storage, there's this new format. It's called OMI-SAR. This is the next generation format. That's why it's called OMI-NGFF. This is basically trying to solve some issues with TIFF. TIFF is restricted to 5 dimensions. This is just how it stores things. TIFF stores always 2D planes and then arranges them in like an internal file system into 5 dimensions by adding like this metadata. But you can imagine there can be experiments where you have images that are very, very, very large. Even the 2D plane is very large. And we had some like where you had 20,000 by 20,000 or something. I imagine something from James Webb Telescope. These are like millions in width and height. So this is then not easy. No computer can handle that. And that's why there is OMI-SAR, which can not only store n-dimensional data, so it can go up to how many dimensions you need, not only 5, but it can also chunk the data, so it basically splits it into n-dimensional chunks. So it basically can also split, if you have a 2D plane, it can also split this 2D plane into like a grid. And then, and also for as a nice addition, it can handle multi-scale. So you have an image where you can zoom. If you zoomed out and don't see all the details, or can't see all the details, it has like a different image that it can show you that is smaller and fits into your memory. And then when you zoom in, it loads basically dynamically the one that it needs. So you have to keep an eye on that one, because these people from OMI-SAR, they are talking with the companies and want to establish it. So you will encounter it at some point. They are currently very much in work in progress, and not all the issues are solved with OMI-SAR, but this is the future, because there are more image modalities where you run at the limits of TIFF very quickly. And then so you're ready for this. So, enough talk about image basics. So, okay, I have some time left. Image analysis basis. Let's switch to image processing and get to the analysis part. So, we are here. When we have our image, we have acquired it. It's saved, hopefully, in some format that is not a blocky JPEG 100 where you see the blocks with your eyes. This would not be good. And then now what we do afterwards is 3 distinct steps, beginning with preprocessing. Preprocessing is basically you apply operations that prepare and enhance the data. This includes, for example, organizing your data. So organizing them in a way that data makes them easily analyzable, organize the conditions, so organize the files so you know which channel order they have. And then also other operations, for example, ones that you're allowed to enhance the contrast, remove the noise that you might see there, remove uneven background, which is quite common, for example, in transmitted light, there's often uneven background, and more. We have a few examples here. Example, you have here these little bit low contrast image, and there are operators that can enhance the contrast so you can really see the details better. Then you have operators that can extract the background, or it's related to remove the background. So, for example, if you are somehow interested in the background here, there is an operator that tries to estimate the background from this image, and then in turn it can also basically subtract this away, so you are with the cells on. Then there are operations that remove the noise. So you see here it's a bit grainy around this cluster of spores, and then the operations that smooth this out, and then it's just the later make your analysis easier. And of course, organizing the data. For example, you have an image that has 5 channels, but you only need 3 because there are 2 other ones where for some tests. You organize them in the pre-processing, maybe remove the ones that you don't need. Then this is followed by analysis, and there you take the pre-processed images that are all organized and denoised, and then you apply so-called segmentation, so segmenting out objects that you want, or you need to do some feature extraction first. You will see what it means, and also do some maybe some machine learning and classification afterwards, and then many other of these operations are available that you basically would classify as analysis operations. And then later on, you do some post-processing. You would collect the results from the analysis and then, for example, filter them. For example, you detected some debris, filter those out. Generate plots, generate statistics, saving everything out. So you have it as Excel file, as visualizations later on that you can upload somewhere or use, and then many more. And this is basically the basic steps of image analysis that are usually never working out. I have to really say this, they never work out. Usually you start with that and then later on you end up with something else, because there might be something that comes from analysis, but it's useful for pre-processing of some data, because you obtain, for example, some cells, and then okay, I need per cell some pre-processing, but I need really to know where these cells are. So this is just a very rough outline to get you started, and then usually each of these image analysis pipelines, they look completely different. So let's start to go through some operations so you build up some knowledge what you can do. So this is in no way complete, but a good start. And you can read up in literature and online, and there are also many tutorials on what is available as operations and different tricks that you can apply. So let's start with image enhancement. So you're basically building up a small toolbox. So when you are working with image analysis software that is not fully automated, anyway, they usually hide it away, usually these commercial software, but more like ImageJ, where you have more control. You have different kinds of operations. Usually ones that modify the image in some way. For example, we have here this contrast enhancement. This is modifying the existing image into enhancing it, but it stays still the same image. Then you have also operations that convert it into something else. It is hard to visualize here. For example, you have here these clusters of spores and converts them into some structure that has this only the information that we need. Or we extract other information from the images. For example, we make an outline around this area and then calculate the average intensity. It's not an image anymore, but we have a value or a table that comes out of that. And there are different operations and how they work, so let's go through them. So first, there's a big difference between operations that are basically working on the whole image. I call it the global operations, and ones that are local, and they work basically on a local window. No, don't worry, I will show exactly how these look. So global image operations, for example, here extracting some image statistics. ImageJ can do this for you very easily. You get a minimum value, maximum value, and some area. So in ImageJ, just as a side note, if you have the area without the selections, basically width times height of the image. And then something local, where you could, for example, calculate around each pixel the local minimum, and then you get something like that out. Is this useful? I don't know, but might be useful for some operations. So, these local operations now. Let's see how they actually work. Usually, these local operations, they have some kind of local window that is predefined. There's usually some parameter that you can change. Example, 5x5 window. And then this window is created around each pixel. And then based on this window, a new value for that pixel location is calculated and then copied into a new image. It's really important. There's but usually the tools will do it for you. For example, I have operation that picks this pixel here. At some point it reaches that one, takes the area around it, calculates something for this pixel here in the center, and then continues on. At some point it will reach another pixel. At some point it will reach here. It will basically do this for the whole image until it's done. This is the basis behind these local operations. And we can use this principle to do useful tasks for us. Example, noise removal. So, what is noise? You can look this up on Wikipedia, you'll find like millions of definitions of these. But one definition of noise is basically signal that we did not want here. There are also different kinds of noise, which makes it a bit more complicated, but common ones are Gaussian noise and salt and pepper noise. And I here took these clusters of spores and then added some Gaussian noise onto it, so some strong Gaussian noise. You will see how it looks like. It follows a Gaussian distribution, that's why it's called Gaussian noise. And it's usually caused by something with the sensor. And then we have here this other noise. It is definitely noise, but it's obviously something different. It's called salt and pepper noise or impulse noise. There are two words for that, basically. It's basically where you have single pixels that are super high or super low, but nothing surrounding it. And this is usually caused by the analog to digital conversion. So I can't remember if I encountered ever salt and pepper myself, but this is how it looks like, and you can also make those in ImageJ and JIPipe yourself to illustrate, to see how these work. So, how can we remove noise with the help of local operations? And a simple principle or approximation could be that we can use is, okay, we have like a pixel value here in the middle, and we have a surrounding area around it. And then we say, okay, noise is a variation around the true signal. So we could take the area around it and use it to estimate the true value from it. So basically we average it out. And this is the principle behind the so-called box filter. That basically goes through each single pixel and then calculates just the average value in its area. And then this is then the new value, the new pixel value. Here's an example of exactly that. I actually meticulously brought out the values here from this image into a table. And it takes all of these values here, calculates the average. Value is now 128. And then this one would be then the new value only for this pixel here at the center. And then this repeats it for all the pixels, basically. And then this way it tries to estimate the noise and then get rid of it, and hopefully it works. Does it work? So let's check here the different noise models that we have, and you'll see immediately how well they work. So for Gaussian noise, we have and the impulse noise, we have the originals here. Then we apply a box filter with a 3x3 area. So it's basically one like that here. It definitely got rid of some noise and suppressed some of it, as you see here, but with increasing window size, you see that this is not legible anymore. I can't even see which cells are which. So this, at some point you can overdo it. And for salt and pepper noise, it's then really funny, because you see here how it then smudges these impulses, and then it makes it worse by applying this denoising, basically. So this is not what we want. So apparently the box filter is not really good for denoising salt and pepper noise. Although it looks very similar in the end when it's 11x11, because I can't see either. And that's why there are different operations that you can apply. There are, for example, under the same principle, that the noise is a variation around the true signal, we can also just use another estimator. Instead of the mean, we can use the median as the local estimator. we instead of calculating the average value here using just the average, we just instead calculate the median. And you see here that in our example last time we had 128 coming out here. Now it's 130 because it's the median value of that. And how does it work? Does it work well? Yes. You see here the clear difference in the salt and pepper noise that it did not pick up on this single one here, which is angering me. But with a 5x5 window, it got rid of the very aggressive salt and pepper noise. So this is nice. And you see some interesting effect also with higher window sizes, is that instead of making everything blurry, so I still cannot distinguish the cells that easily anymore, but it's the contours are still sharp. That's just how the median filter works. It rather extends the bright parts into, makes them larger, but it keeps the contours usually very sharp. And then that's why it's often very used. Sometimes the box filter is also useful. It really depends on what you want. And there's also another idea what we can do. So the box filter has dumb assumption action. Basically, when we take a local area to calculate the average, it makes an implicit assumption that each pixel's basic contribute are basically of the same structure. What do you think if this pixel that is very close to the center that we calculate is how likely is it that it's that this one is here contributing to the same structure as that one here. So this one here is very far away, and this one is closer. Yet in the calculations, they contribute the same in the value estimation, and that's why we should really weigh them. And there's one filter that will do this for you. It's called the Gaussian filter. The Gaussian filter is basically making a instead of weighing all the values the same, it's basically weighing the center the highest, because this is basically our center point, and then it decreases the further away we get. And it's called Gaussian filter because this distribution here is basically to the Gaussian distribution. There are also 3D variants of it which make a 3D Gaussian distribution. And then how it will work is basically when we do the calculation for our center point. So I also wrote down these pixel values, which was very annoying. And this filter kernel, how it's called now when we have weights in there, and we basically calculate this. So we calculate 15 times 0 plus 220 times 0, 0.1 times 154. We do it for all of that, and then we that way we can calculate by summarizing everything the final value that comes out for this single pixel. This is basically how all of these local operations now work. They actually have weights inside. It's very, very powerful. And you can imagine is that you can change here these weights. You can have, of course, a different size of the window, and you can change the weights to something, maybe something useful. And there are many, many of these filters that are having different weights inside here that do very, very useful tasks. So let's see how the Gaussian blur performs. It's similar to the box filter. So it blurs everything to hell. And now here it's sigma, because it is basically using the Gaussian sigma as its main parameter. It cannot still handle salt and pepper noise very well, but you see here that if you compare this a bit, then it's a less blocky. So I think I have here a comparison with everything. So yes, I have here a comparison with everything on the Gaussian noise image. And you see that the box filter and the Gaussian blur, they look very similar, although they have some blockiness. If you are closer, like in here, you see something with the box filter. This actually has some mathematical background to why it is like that. You can ask me later why. And the median filter, of course, performs quite well. and keeps these structures sharp in the end. And then for impulse noise, it's king, because it absolutely gets rid of these impulse noises without any issues, unlike the other two others. And then, as mentioned before, it really depends on what filter is useful for what. You can also use them for something else except denoising. You will see some of these examples in the exercise, I think. Also, that might explain some. So, next image enhancement. Let's see. Oh, time good. Let's see about another tool that you can use for image enhancement. It's histogram equalization, and this is related to contrast. So contrast, there are like millions definitions again. What is the contrast? I used here the Michelson contrast, and it's basically that we want to make use of the whole value range. And this is why it was so important to talk about bit depth and the ranges that are available, because the bit depth of the image and what data types you have, they determine what min and max signal you have available inside of each pixel. And we want to make use of this. This is the idea behind it. We want to make this super dark image into something that we can actually see. And we can actually see the low contrast condition in the histogram of an image. So if you can see here, I actually mapped this out into a histogram with image shape. And you see that we only use from 8 to 63 as the values, but we have a range from 0 to 255. So we should really correct for that so we can see more of the image. And there's a simple tool to do this called histogram stretching. You basically literally take that and then, like in Office, when you stretch something larger, you stretch it. There are two variants of it, min and max stretching. Basically, you then would shift the 8 here to 0, drag it also from the left, and also do it to the right, so we max out the full value range. Or we only do the max stretching, so you would only grab it from the right and then max that one out, but leave the left side alone, because there might be some information in there, or it could be used so that this one is left untouched. In there are many operations that are available that would actually apply the histogram stretching for you. And now, what is histogram equalization? So this can be mixed up when you are unfamiliar with those. Histogram stretching is very, very simple operation. But it generally does not enhance the image that much. So it is basically something that makes it easier to see. But there's actually another method. It's called histogram equalization, and this one is a bit more advanced. The histogram equalization actually is a complicated operation, and I had to prove once the mathematics behind it, which was fun. It's the goal is to make the histogram uniformly distributed, make it as uniformly distributed as possible. So we make use of the whole range, but then also distribute the values in a better way. Many of these tools that implement histogram equalization, they also have some additional settings. Image has like a percent saturation that you will see in the tool. There you will do this when you're using when you're doing the exercise 1, you will actually use this histogram equalization from ImageJ. And you see here as output that it's actually not uniformly distributed. So you see here the stretched histogram, and then the supposedly uniformly distributed equalized histogram. Why is that? Because we have discrete values here, and it just doesn't work. The histogram equalization mathematically would make a uniform distribution, but it assumes continuous space. We don't have this here. So don't be surprised if it doesn't look super uniform when you use actually this method. It is just limited by its by the mathematics and how computer versus mathematics. So let's try and see what happens. So here's the original image. You see here this histogram that only goes to 63. So if I apply the histogram stretching with the ImageJ method that it is available, then it looks like that. So it's stretched out. And then with the equalization, it looks different now. So it has distributed some of these pixel values a bit differently. So they are now super bright. So in our case, this one is not maybe super helpful because it enhanced this noise to the maximum that is surrounding here that we could handle maybe fine before, but now not anymore. So there is no one solution fits all. It's like maybe does the stretching work, maybe does this equalization work. And we have one example where equalization works really, really nice and enhances something that we need in a very low contrast image. So really try it out. That's why I show you multiple operations. Then there are also images that are a bit annoying to handle, contrast-wise, that are non-uniform contrast. So you see here this part, this is from a TEM image. These parts here are low contrast because there is this, there's like a rip or whatever, that is very high contrast. And this would totally mess with our analysis because this one here is so high contrast that it would be detected so easily by each algorithm. So how do we solve that? There is fortunately an algorithm for that. It's Klare. You will usually find this in many of these tools. Klare means contrast limited local adaptive histogram equalization. So it basically, the idea behind Klare is it applies a local area to find the local contrast, but then adds some additional calculations that are very useful that prevent it making parts of the image super high contrast that shouldn't be. So like there's a system method that is R here without CL, and then this one has this problem. But Klare works fine, and you see here in this image now it's uniformly contrast. The annoying whatever artifact here is the same, and then in the following analysis will be now easier. And this is actually a very, very, very common principle. There are many algorithms that you can find that apply such local operations and are smart. For example, there's a local adaptive Gaussian filter, adaptive median, even some image processing tools like Photoshop, they have like adaptive Gaussian filter that blurs everything that has lowest information, but then everything that has structure is left alone. It's very, very useful for some image processing tool, not only for analyzing scientific images. So what do we have now already? We have something to remove noise. We have something to enhance contrast as well, different tools that are available that we can use for it. Now, thresholding.
Excuse me.
Yes.
I'm just wondering for the histogram stretching. When we are stretching, does it mean that we are also increasing the bits, or it's not related?
So basically the bits are basically the whole area that is available. It's predetermined because it's part of the image itself.
So it was already there, but actually we weren't using it.
But we don't make use of it, basically. So you see here, this is my whole, like my boxes that I have available, but then I only fill only 1/3 of my boxes. That's basically. And the histogram stretching basically on the histogram equalization tool, the idea is to make use of your whole range that you have available. So later on, the algorithms respond better, you can see it better too. You imagine here you have something so low contrast, you can stretch it or equalize it so you can actually properly work with it. And we have actually some example data where you absolutely need these because it's so low contrast, especially the 16-bit images, they go to 65,000 something, but then our structures are like at 1000. And you don't see stuff anymore. You have to change it in the contrast tool, which does the same thing when you drag it, the slider in the image editor. It does exactly that in the background for you. So you can see it. And then this would do it programmatically. So our following algorithms that we want to work with for the analysis actually see it. Well, we can't see it and the tool can't see it. That's basically the idea. So, thresholding is the next thing. Thresholding is very, very common. We will encounter very different variants of these thresholding operations all over. It's basically converting something that is a grayscale image or a color image into something that is a binary image, so black and white. I have to note here that binary images are theoretically 1 bit, because 0, 1. No computer works on 1 bit. The minimum a computer can address or work on is 8 bits, so 0 to 255. So usually in these implementations, they are represented as 8-bit image, where then it's okay. If it's larger than 0, then it's segmented out, or it's thresholded. So here's, I've written it down again: background, everything that is value 0, foreground, everything that is not 0, or larger than 0. And we don't go below 0 because they are integer numbers and they start at 0. So I thresholded here the cluster so you can see one example. So the straightforward thresholding is doing it manually. Each tool has their interfaces for that, JIPipe as well. So you go, for example, image, you go into image adjust threshold, and you can change some sliders. It will show usually what it will find. Then you tell it, okay, do it now, and then get your threshold out. This works. Is it good? This is a different question. Because you will run quickly into some issues with this on manual thresholding. You can also threshold colors. So as an example, find the banana. This is difficult with RGB thresholding. So I have here a 2D image that is RGB colored, and this is not possible so easily, because where is the yellow here in the set? Yellow in RGB is a mix out of red and green, so you sit there and try to figure out yellow. So this is a bit hard. That's why I mentioned a bit these color spaces in the beginning, because they become now useful when you want to do manual thresholding based on colors. And there are two very common ones that many tools implement. It's HSB, hue, saturation, brightness. It's usually the one in the image editor where you can switch to. But then one that is less common in image editing tools, but more scientific, called thilo. JIPipe will do the conversion and for you easily with some operations into that. So then you have basically a different interface for the thresholding. So here you can pick out the yellow, for example, very easily, and then make it a bit more saturated, and then nice. And then for thilo, you can pick out the yellow literally here from its thresholding component. And here I tried my best to match the banana a bit. It mostly worked, but then you see also that this weird thilo space that is very uncommon worked actually better. Was a little easier, if I remember correctly, to somewhat get a good classification out of that here. That's why I suggested it to some software developers as well. They should implement this thilo space. So you have just seen it how this color thresholding is. But color thresholding in general is relatively rare. What usually thresholding is just black and white images. And there we are back with the problem. You're sitting there and working on some manually changed setting slider and then try to match it up somehow. But we can totally automatize it, because there are actually tools that would calculate an auto threshold based on different algorithms. There are many in ImageJ, for example. You find auto thresholding and intermodes and Yaga and Huang and so on. There's like 15 of them in ImageJ. And they're very useful, and they would do some kind of thresholding for you automatically based on mathematical criteria. So you don't have to sit down and threshold stuff out. The most common one that everyone also explains, including me now, is auto thresholding. It's basically like all of those actually. They look just at the histogram. Basically in this image that I stole from Wikipedia, you can see how it works. It's basically goes through the histogram and then applies a thresholding. Okay, from the left side this is the background, to the right side this is the foreground, and it basically says, okay, what is the maximum? What is the point here, and where our threshold is, where the variance between the classes, so the background and foreground pixels, is maximum. Then it would pick this one here out. This is basically how OTSU works. Very straightforward, that's why everyone explains it. The other ones, they are based on some entropy measurements. So it's very, very useful for workloads where you have a very clear bimodal distribution when you look at the histogram. But it works somewhere else. You just have to try out how the threshold works. ImageJ, you have like a try all node, and then JIPipe I made one for you that you can find somewhere in the board. I will mention it to you when it's time. That would try out all the thresholding methods, and usually you pick one that is the best. You will get a feel for how to which these thresholding methods react very well. There are also variants for local thresholding as well. And straightforward would be like local OTSU, or local contrast threshold, or local whatever. This basically goes through each window, then calculates the threshold for OTSU, for example, in this local window. And this is the one threshold, and for this pixel, repeats everything for all the pixels, just as before. But then there are also smarter ones. So if you have these one here, Niblak and Berndsen thresholding, you will find them both in ImageJ and JIPipe. These are extremely useful if your contrast enhancement methods could not handle it properly with getting the contrast right. So you might have like structures that are a bit dim, and then ones that are a bit very bright. And then these local thresholding methods, they can usually handle them fine because they look in the area around themselves. They are a bit smarter than this local OTSU and local contrast threshold. They would do some testing if it's actually making sense. So it would, for example, exclude areas that are very, very dark and there's nothing there. Or less likely that there's nothing there. More likely there's nothing there. So keep these here in mind if you are struggling with your thresholding and it does not react or reacts unevenly to your image. So 1517, this one I can quickly do. Let's talk about feature enhancement a bit. So what is a feature? So you have here an image, again, these clusters that I always used. And a feature is basically a region that has some kind of properties that are of interest. There are different features available. And so far, for our thresholding, we used a simple feature. Okay, it's everywhere where it's bright. This will not always be sufficient. You will easily break that. There are different data sets that just don't have this. So we need something that reacts well to different kinds of things that you are interested in. And I have here 4 of these commonly used or commonly encountered features that you will find in images listed. So ridges, blobs, edges, and rough areas, or texture-based areas. And I will go through them one by one, so don't worry about this overview. So let's start with. Why do I start? Why? So as we are now here, should have a small break. I think like 5 to 10 minutes or so. And give my voice also some time to recover. So, everyone ready? Give my recording still works. Okay, continues with binary processing now. So let's assume we have thresholded our image, and usually it will never be perfect, what you have found. Example with these clusters, you see that these one have a hole, you might want to have it filled. Then you have often, especially with these biological images, things that are absolutely merged together. So here maybe I can assume somehow how they are split. I tried it. And for the cat with the banana, you have here wrong these segmented parts here. Also the flower in the middle was the yellow part was detected. You have disconnected parts here, actually quite difficult to handle when they're disconnected. But we have to process them somehow and move the ones that we don't want and fix the ones that we want. So, and for a main or very useful tool that you can use on it are so-called morphological operations. So these I mentioned already with this morphological top hat filter, but now let's talk about it a bit more in detail. So morphological operations, if you look them up in literature, you get blasted with math or some weird example. But basically what these are, and it's just the easiest way to think about them, is they're just local minimum and local maximum operations. And there are two basic ones, the local maximum and the local minimum. And they are called in historically, and because they are mathematically something else, sets something mathematical, they're called dilation and erosion. And it's obvious why they're called like that. We have here this original image, this I stole from OpenCV's examples. If you apply the dilation, so a local maximum operation, then it gets dilated. So the image, the mask gets all enlarged, because you can imagine when we are here close by nearby at a white area, but on our current pixel is black. If we have a surrounding, if we take a local area around it and it hits even one pixel only that is white, then the current pixel will be made white, so everything inflates a bit. The opposite operation is erosion, and you see here what happens if we apply an erosion operation. So, and these can be then combined into higher order operations. The ones you have seen already, only a bit. It's specifically the opening and closing operation. The opening we've already used. This is the erosion followed by dilation. You see what happens here on the examples. Opening, basically you can imagine it as it opens holes. So it opens parts that are small and basically removes them. It erodes the image first, so it gets here rid of the smaller dots. But then it would also, as you see here with this erosion, thin out the J. But then because you dilate it, then again, ideally with the same radius, then it basically restores the original, almost. It's never restoring it completely, but at least for larger radii, but close, very close. Then you have its opposite operation, it's morphological closing. And this one is easy to explain, it's basically you close holes. We have here these holes, small little holes inside. The dilation operation would close them all, because it's a local maximum. And then it would dilate it. So we undo it by applying erosion afterwards, and then we get our original back. You have to be, of course, careful with the radius, because you can configure them. You can set the local area around it, and also like a structure element. If it's like a disk element, and it would round off the objects a bit, usually more useful for biological data. But you can imagine then if it's connected like here in this dilation image, this is not going to disconnect anymore after that. So you can accidentally connect things with the closing operation that should not be together if you set it up wrong. So be careful with that. And then splitting them up again is really fun. Not. Then you have some also funny ones. The gradient operation is very useful if you want to make a mask that has like an area into an outline. And the top hat, here you see why it's not so used so often. We have seen actually what the top hat does with the glomeruli. It's extremely good at highlighting blobs and ridges. This example is super not useful at all. So see it as local minimum, local maximum. It opens up more possibilities than binary on what it usually is defined on. You can use this gradient. It's very useful for visualization, for example. You can comment it for that. Then there are also other binary operations that are not part of this set of morphological operations. They are a very distinct set, but also sometimes called morphological operations because they are changing the shape of the objects. We have like a hole closer, but a different kind one. Then object separation filters, and then general object filtering that is all implemented in different tools in a different way. So let's look at fill holes. So the hole closing has, of course, as you've seen, the dependence on the radius, and sometimes these holes are super large, and you would basically destroy your object if you increase the radius too much. But there's another operation that you can use. It's the morphological hole filling. It's called different in every tool. It's inconsistent naming. In JIPipe, it's called morphological hole filling. Some tools call it flat fill, some tools call it fill holes. I've seen basically all variants of this already. You see here its benefits already. Morphological closing. I have to increase the radius so much to close finally these holes. All the other radii were not enough. But the flat fill operator is very nicely working. So how does it work? Well, it's basically doing the same as the paint bucket when you go into MS Paint and you use that one. Because basically you take the original image, then it applies it in a paint bucket outside here, somewhere at the border, and in very specific pixels, namely the one that are white, but after inverting the image. Then you basically get only the inner parts out, and then basically combines these two images by adding them up. So it basically makes this here all black, then only these inner parts are left over. We have the inner parts, the outer parts, add them together by pixel-wise adding them. Boom, we have the holes filled. This, of course, has one disadvantage. It has, if we applied some operation or our structures are just like that they have holes that are completely disconnected from the border, from the image border. So if you imagine if there's something in between, so this one here is completely separate, then this one also will be filled. So it's not an all-in-one solution. Sometimes there are no good ones, and then you have to do something or some more advanced operation. But it's usually a good start. Then I want to show you also a very, very useful algorithm that is useful for biological data. It's combined smoothen and fill operations. So these here are from the kidney data. It's actually zoomed out a bit. So far I showed you only the glomeruli, but this is actually from a study where these clearing methods were showcased for light sheet. It was light sheet data here. And there they wanted to compare the tissue sizes as well. And then, so they needed to do the segmented tissues. Simple auto thresholding gets you that here. So, but we have these annoying holes inside. Okay, we can close them with hole filling. But then you have these borders here that are a bit unclean. They are due to the issues with the segmentation. So there's a simple protocol to basically solve both. Also, there were images where I didn't have it here, where parts were not detected properly, and then the hole filling could flow basically inside because there was a path to the inside. So there's a simple protocol to solve it. We first take our object or our mask, we dilate everything. You see here that it did not close every hole, but then we apply the hole filling, and then we erode it afterwards, so we get our original size back. And thanks to the morphological dilation, we basically cleared off here this part. I don't know if I show it to you. No, I don't show it. But if you look closely, then it's nice and smooth then afterwards, because the morphological dilation has basically smoothed all these parts nicely off, and then the holes are filled too. So this is what we want in the end. So then cluster separation. Cluster separation is unthankful task for biological data, and it's not so easy to solve, unfortunately. So if you have ever something in test where you have clusters, then pray that the simple one works. And the simple one is so-called distance transform watershed. Here, if you have a cluster separation in general, the difficulty is that these algorithms, they struggle with, okay, what is an object? So if you look at it, just at this mask, you might infer in your head, okay, where's the border? Maybe like here, and then here, and then here. But the algorithm is maybe a bit dumb and doesn't see that. It needs some criterion that is hard and can be calculated to find out the objects. This distance transform watershed algorithm basically is it's a combination of two terms, distance transform and watershed. But it applies, I don't show it here unfortunately, but I can show you maybe soon because I'll be finished faster than expected. It applies a so-called distance transform, where each of these white mask pixels here basically is reassigned to a new value that corresponds to the distance to the closest black pixel. So it's basically then you get a nice, an interesting looking image that basically contains the distance information. It's very, very useful for many, many things. And there it would basically pick the local maxima out, which hopefully correspond to the centers of your objects, and then watershed them. So watershed is basically a method to do segmentation based on given seeds. So they basically give, okay, there's the object, there's the object, there's the object, and then find a way to segment them so it fits the contours. And distance transform watershed basically does this all in one step, finds the centers of your objects and then separates them, and you get something like that out. It worked somewhat. So it worked fine for these, but then here I would see 3 cells that are still clustered together. So it's not always successful, and the only thing that was successful that solves it very consistently, I think, is AI-based methods. So something like cell pose segmentation can usually solve clusters very well, but they have a very complicated setup there as well, and you need more computational resources for cell pose. But then you get a good clustering out. We might find the time to maybe try it out during the workshop. We'll try out cell pose, but I don't plan it with cell pose because it's quite heavy on the hardware. So then we are stuck with this way too large object and we can't get rid of it. So we have to filter them out. And there's also just an important part, you need object filters or selecting specific things. And unfortunately, for example, a criteria, for example, area, brightness, whatever features, circularity, location. The problem is every tool does it completely differently. Imaris does it different to ImageJ, does it different to OpenCV, does it different to whatever Python uses, the SK image. Every tool does it differently, calls it differently, and the algorithms are called differently as well. So we learned it for JIPipe and ImageJ, so they are very related to each other. And here they are usually working with regions of interest, how they're called.